Biological Studies and Professor of Biological Sciences at the University of California, San Diego. He's a fellow of the Institute for Electrical and Electronics Engineers and received the Neural Network Pioneer Award in 2002, and in 2003 was elected to the Johns Hopkins Society of Scholars. Dr. Sejnowski founded Neural Computation, published by MIT Press, the leading journal in neural networks. He has all sorts of accomplishments, and I don't think I should hold you up. There's a whole page of excellent things about him, and he's going to talk to us about wiretapping the brain. Very Sejnowski.
Good. Thank you, Fred, and thank you all for coming. This coming back to Hopkins brings back memories. I remember when I joined the faculty back in 1982, started my lab, I remember when they renovated Jenkins, because we had to move out and camp for a couple of years in Mergenthaler, the old biology building. This is before your time. But I remember how disruptive it was. So I really feel for the people from Barton. I know what you're going through. It's going to be better, but you have to be patient. So, in fact, the topic of today's talk is one that really goes back to those early days here at Hopkins. This was a slide from those days that I resurrected, which I used to illustrate the many different levels of investigation that one can take when trying to understand something as complicated as the brain, which arguably is the most complex device in the known universe. Now, I always thought that, but my wife was a physician, pointed out to me that, in fact, it's not, that, in fact, the body is more complicated than the brain, because the brain is only a small part of the body. What's challenging here is that there's important structure, spatial structure at levels of investigation that range over 9 orders of magnitude, from molecules down here at the very bottom to the entire central nervous system. And neuroscientists over the last few decades have systematically gone after each one of these levels, trying to understand and work out all the molecules, for example, that are used by synapses to signal between neurons, and all the neurons themselves, many different types. And molecular techniques have been, genetic techniques have been incredibly important. And they've left us, the molecular geneticists have left us with an incredible puzzle. I call it the Humpty Dumpty problem. So you remember that all the king's horses and all the king's men couldn't put Humpty Dumpty back together again. And that's what basically neuroscientists have done. They've taken apart the brain into its component parts. So we now have a parts list. It's by no means complete yet, but we have wiring diagrams. We have a sense for how the time courses for signals and so forth. But the mystery that still exists, and the challenge, is try to put it all back together again. How is it that we're going to understand how the brain solves such problems, for example, as speech recognition? If it wasn't for the fact that human beings can actually understand speech, I'm not sure anybody would ever think to try to automate it, right? Would it even be feasible? So it's an existence proof that a lot of difficult computational problems can be solved. Nature has solved them in its own way. Unfortunately, the type of engineering that nature has used is alien to us. We really don't know the basic design principles. In fact, it's probably a mistake to think about it in terms of design, because the brain got to where it is by evolution. So what we really need to do is to look at the brain and look for hints, things that will help us to understand what are some of the principles that the brain, that nature used in order to create something that not just works and solves all these difficult computational problems, but somehow managed to do that through this process of gradual changing of the genes in evolution. So here's a hint. This was a paper that was published by Ketchen Zhang from my lab when he was a postdoc. He's now on the faculty in BME, and he might even be here. He's not here? Oh, well. Well, I know that Murray Sachs is here, and Murray Sachs will get this back to him.
The fact that he's not here, you mean?
Well, no, you could tell him that I showed one of his slides. Okay. This was a study of scaling. So how is it that, for example, we have the brain of a pygmy shrew that weighs a gram, and we have the brain of the elephant that weighs several kilograms, and they all use the same plan. These are all mammals, and they all use the same basic anatomical structure, right, the mammalian brain. Some parts are bigger than others, but pretty much the ground plan is the same. So what is plotted here is specifically regarding the size of the cortex. The cortex has two parts. There's the gray matter, which is the 2 or 3 millimeter thick rind on top of the surface, which is deeply folded, containing neurons, synapses, a very complex meshwork of interacting neurons. So that's the gray matter, and you can look at the volume of the gray matter in all these species. And here's the other half, which is often ignored, and that's the fact that underneath the gray matter are a lot of wires connecting different parts of the cortex and projections down to the subcortical, back and forth between the cortex and the subcortical structures, called the white matter, right? The white matter, it's white because it's myelinated. It's covered with a fatty insulator, and therefore it looks white. It's easy to distinguish from the gray matter. So here's log white matter, log gray matter. And what's amazing is that despite, again, over 6 orders of magnitude of spatial scale, scaling up this basic body, this basic brain plan, we can get a correlation coefficient that is 0.998. And I can assure you that it's very rare to get anything that accurate in biology. Biology is really something that tends to be very highly variable. Things aren't manufactured to spec. Things come in many different sizes and shapes, and somehow it adapts. But here seems to be a scaling law. And furthermore, and here's the real hint. Okay, this tells us that there's a principle here, a scaling principle that allows us to go from a small, little miniature computer to a giant computer in terms of numbers of neurons and sizes and so forth, and it's done with the scaling law. Look at the scaling coefficient here on the log scale. The slope here, 1.23, tells us that the white matter is actually growing faster. The volume of the white matter is growing faster than the gray matter. As you get bigger brains, you have to put in proportionately more wire, roughly as the 4/3 power. 1.23 power. So that's telling us that wires are really important. And those wires are ones that, by and large, connect distant parts of the cortex. These wires, although there's a lot of them, it turns out that the cortex is very sparsely connected. If you take a pair of, say, a neuron in the front of the brain, the back of the brain, the probability there's a direct connection is like 1 in a million. But even within a small column of the cortex, say within a millimeter, where there's a much higher degree of connectivity, the probability there is still only 1/10. It's higher, but the question is, how do you organize a very complex system where information is stored all over the place and decisions have to be made very rapidly to, for example, if you recognize a tiger, that you should run in this direction and not that direction, or that someone is calling you from behind, so you have to know who it is, that recognize who's calling you and where the information is coming from. All those decisions we're making instantly within hundreds of milliseconds with a system that has a very slow clock time. The clock time of the brain is measured in milliseconds, right? So you talk about gigahertz, right? This is off by a million in terms of the clock speed. So we have a millisecond sort of time scale for the single device, for the neurons, but we have a lot of them, and we have an incredibly distributed memory network and processing network, and somehow it's all got to come together to solve problems very rapidly and efficiently. That's the mystery. So that's what we're going to try to explore today. So, so this story begins back in the, let's see, I was going to say the last century, back in roughly 1970. And Simon Laughlin is an invertebrate biologist at the University of Cambridge. He was studying insect eyes. And he asked the following question, okay? So I want to transmit information from the visual world into the brain of this insect, and I want to maximize the flow of information. Now, I know from just looking at the distribution of light intensities over the natural scene that if you think of X as the intensity of light on the photoreceptor, this is the output of the photoreceptor. So it has some sort of variance. The light goes from light to dark, and then there's some sort of peak here. And now the photoreceptor has to transmit that information to the second order neuron, right, through a synapse. And the question is, what would be the optimal transformation, nonlinear transformation, in order to maximize the flow of information through that synapse? So here's the nonlinear transformation. This tells us for any given output from the photoreceptor, this is the output you should give to that neuron, right, the output of the synapse. And the answer, he was able to answer this question in a very elegant way and actually show that the insect was adapted and actually had a synapse that looked like this. The answer is that you need to have a nonlinearity that optimizes or maximizes the entropy of the output distribution so that all possible values are used equally well. So you use the whole, in a sense, the whole channel bandwidth. So how do I make this output as flat as possible? Well, the way to do it is to, first of all, take the peak of this and put this at the center of the sigmoid, and then to adjust the slope of the sigmoid at the midpoint so that it matches the variance of the input. This has to be adapted because depending on where the insect goes, it may have a distribution in the shade which is narrower or wider. So there has to be ways of online adapting this transformation. But the key insight here is that we need to be able to adapt. We need to online to figure out where the mean is and adapt, and then also know the variance. Online adapt those two parameters. And that basic principle turns out to be key to solving another difficult information processing problem, which is called the blind source separation problem, or the cocktail party effect. So here, imagine people talking in a room. And imagine that we simply take, first of all, an individual, a single source, a single independent person, and simply record the speech from that person and plot the values of the plus and minus signals, the pressure signals in the air. And I want you to compare this distribution with a Gaussian distribution with about the same number of samples. There's noise, fluctuations, but I think it's easy to see that these two distributions are radically different. The Gaussian distribution has a much flatter top, it's rounder. This has a much more peaked top, which means that there are much more zeros here than in a Gaussian distribution. And these tails here are also much longer. So this would be said to have a high kurtosis, higher order moments than the Gaussian. Interestingly, almost all natural signals that carry meaningful information, fall on the left here. For example, even rock music, and some would argue about the meaningfulness of the information. But music in general is also super Gaussian. And if you analyze images, for example, image intensities across natural images, they're also super Gaussian. So what it's telling us is that nature, where there's actually information embedded in that signal, often will give you these long tails and lots of zeros. That's just a fact. So here now is the cocktail party problem. Okay, suppose that we have a bunch of people. Let's call them speakers S. And here we have 5 people, and they're distributed around a room. And we have 5 microphones, X, picking up the sum of the pressure waves from all of the people who are talking simultaneously to each other. So 5 mixtures, and let's assume to begin with that there's a linear mixing matrix. This A can be generalized. We can put in time delays, for example. We know that there's a finite travel time for sound in the air, and which is variable from speaker to microphone. And also, you can imagine the acoustics of the room might be important. So in general, you might want to put an FIR filter in here. It turns out that all of those generalizations can be encompassed within this general framework. So let's just start with the simplest case, which is a linear transformation between S and x. Okay, well, it's a linear transformation. It should be possible to find the inverse of that matrix as long as it's not degenerate. And so the goal of blind source separation is to find a matrix W which inverts A such that the transformation to the output here, the U's, recovers each of these speakers individually. Without knowing, it's blind, without knowing any of the characteristics of the speakers. Or in fact, whether it's a person talking or it's music. In fact, in order to solve this problem in the most general form, we should be able to solve it when each of the so-called speakers is actually a white noise source, right? A microphone, X, is going to pick up a sum of 5 white noise sources, which sounds like white noise. It sounds like you've really tied your hands behind your back. You've got nothing to work with. This same framework, by the way, can be used for designing blind deconvolution algorithms, where now, instead of 5 spatial locations, you take 5 time taps into the past, and you're trying to find some way of extracting independent components that are functions of time. So this is a very general framework. So, so here's the formal statement of the, first of all, the source model. Here's the matrix A that's linearly mixing the sources. And here's the output U, which is an unmixing matrix W, which inverts A. And now we're going to make one assumption. And this assumption turns out to be the only one, actually one of 2 that you need to make. You have to assume that the sources are statistically independent. That is to say, the joint probability distributions will be simply the product of the individual source distributions. Now, this is a much higher and much more strict criterion than saying that the signals are decorrelated. 2 signals that are decorrelated means that they have 0 correlation. That's a second-order moment. If they're independent, it means not just their second, but the third, fourth, and all the higher-order moments have to be 0, too. Now, the second condition, it turns out, that you need in order to be able to solve this problem is that you have to assume that these probability distributions for the sources are not Gaussian. Now, this is almost the antithesis of the approach that is normally taken when signal processing analysts try to attack a problem. I remember that when I was a physicist a long time ago at Princeton, when before I went to sleep, I said a prayer to God. I said, Lord, let the world be linear, Gaussian, and let the signals be stationary. And the reason why you pray is that those are the only circumstances under which you can actually analyze and come up with an analytical solution to the problem. You know, there are occasionally nonlinear problems you can solve if you're lucky. But we're dealing here with the brain is dealing with signals that are not Gaussian, speech. They're dealing with high degrees of nonlinearity. These neurons are very nonlinear devices, and there's all kinds of nonlinearities in the system. And it sure is non-stationary, right? There are changes that are occurring in terms of the state all the time. Somehow the brain has to track that. How does it do it? So I'm just going to outline the approach that was taken by Tony Bell, who's a postdoc at the time, who had just finished his PhD in computer science. And he was interested in generalizing Simon Laughlin's insight. He said, well, if it worked for one channel, maybe it would work for n channels. So what I'm going to do is I'm going to take the entropy of the output, of all those outputs. I'm going to try to maximize them. Well, first of all, he proved a theorem. He proved that if you maximize the entropy of the output, you minimize the mutual information between the outputs. And that's taking you toward independence. And then what he discovered was that if you used a sigmoid, and here's the function, it's the traditional sigmoid function, 1 over 1 plus e to the minus u, and use that. There's just 2 parameters here. Remember, there's the kind of the sliding mean for the threshold, and then there's the slopes. There's 2 parameters here that if you use that function, you could come up with a closed form iterative solution to the blind source separation problem. And here it is. Here's the algorithm. Here's the unmixing matrix W, and it says that at every time point, what you do is you accumulate some information about the outputs U, and then multiply this, which is a linear sum of the weights and those outputs, by a function here, which is simply the sigmoid, right, by this nonlinear sigmoid, and add in a little bit of the previous value of that weight, and iterate on every time you get a new piece of data, simply iterate that. And what he showed was that for a very wide range of inputs, including all the natural ones, speech, vision, and so forth, that we looked at, this one nonlinearity worked. It turns out that in order to prove that it's going to converge, this function here has to be the integral of the probability distribution of the signal. That is to say, it has to be the integrated probability distribution. But for super Gaussian signals, it turns out that this is a good approximation to that. Okay, so I'm going to give you a demonstration of this, I hope, which will illustrate it. In the case, this is a real demo in the sense that it was 2 microphones and 2 people talking, or actually one is music and one is a person talking, in a real room, and we're using a version of the algorithm that Tae-Won Lee has developed that includes time delays and FIR filtering. So this is really a real-world, real-time problem that we're trying to solve. Here are the 2 mixtures in the microphone 1 and 2, and here is the final source solutions. And now, I'm not sure if this is going to work, but we're going to try to play and see if the yeah, looks like this is, put up the volume. Okay. Okay, so first let's listen to the sources, the mixtures.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
That's mixture number 1. Mixture number 2.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
Actually, I wonder if that was the right one. Let's see. Let's try that one again.
1, 2,
Oh, this is the second mixture.
4, 5, 6, 7, 8, 9, 10.
Okay, so that's the input to the algorithm. The algorithm iterates. It's not like PCA where you can just write down, just do the eigenvalues of matrix. You have to actually go through and gradually, through gradient descent, achieve a stable asymptotic solution. And this is, at the end, this is what it sounds like. This is one of the outputs. Again, it's completely blind.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
So you can hear there's a little bit in the background. It has about 25 dB separation. Well, that's nice. The algorithm actually works. And it really was, in a sense, at the time, a breakthrough because this whole problem had only been formulated about 5 years before by a French group. The idea that you could use independence as a very strong criterion for separation was first developed by a French group. There's Pierre Comment, who formulated the problem in the modern version. And the first algorithm that had ever been developed, which worked sporadically under some circumstances, was developed by another 2 Frenchmen, Hérault and Jutin. This particular algorithm, which is based on information theory, it turns out to scale exceptionally well. That is to say, you see it here for 2, but I can increase this up to 100. And the algorithm obviously takes more time, but it produces very good separations.
How long did it take to do this?
This is like a millisecond. In fact, Tae-Won has versions of this now that work in real time. And he has a company which is now marketing this to the military for use inside of tanks. One of the major problems that people have in tanks is being able to communicate with other people in the tanks because the huge background noise. You're sitting on top of a 1,000 horsepower motor, and there's banging all over the place as you're moving along. So getting rid of the background is a signal blind source separation problem. And so being able to solve that means you can immediately get rid of all the background noise and just concentrate on the smaller signal. Yeah.
You mentioned something about the sources not being Gaussian, otherwise I can see how you can't separate them.
Right.
I also noticed when hearing the last channel, the music,
Right.
that I could hear the S of the 6 and the 7
Yes.
much more clearly.
Yeah. So because of the fact that there are going to be reflections off of the room, okay? If you don't model the reflections, then there's going to be a residual. So what you were hearing was the fact that it wasn't a perfect separation.
So it's not because the S's tend to be much more white noise?
No, I think it probably has more to do with your ear is more sensitive to the S's. But the fact is that at the end of the day, you could quantify how well the separation is in terms of dB. And that may be different from acoustic. In other words, how you report perceptually, whether it's a good separation or not. Okay, so what's going on here? There's a geometric way, a simple geometric way of just looking at this, which gives us an insight. Okay, let's suppose we have just two channels. Speaker, the microphone 1 and microphone 2, and these are the data points, right? Because there's two mixtures. You get two limbs here that are kind of like a cross pattern. Let's suppose that we wanted to use PCA, principal component analysis, to do the separation. What PCA does is it looks for the direction of maximum variance. And in that case, for this case, the direction of maximum variance is right along this line between those two limbs. And PCA requires that all of the directions are orthogonal. So PCA, remember, is just looking for decorrelating using second-order moments. And here's the second direction that's orthogonal. You see there's no data in that direction. So what PCA is doing is actually mixing the two sources, and it's doing exactly the wrong thing. ICA doesn't make any assumptions about orthogonal, what direction the axes are going. It doesn't have to be orthogonal. And as a consequence, by looking for directions of maximum independence, it discovers this. Now, this is two-dimensional space. If you have n channels, if you have n microphones, then you're doing the same thing in an n-dimensional space. So another way of thinking about this, if you're familiar with projection pursuit, this is a multivariate version of projection pursuit in which you are simultaneously pursuing n dimensions at the same time. Richard?
N speakers, not N microphones.
Yeah, n microphones. Okay, I should have mentioned that you have to have n mixtures. If you have n speakers and n sources, you have to have n microphones in order to do the separation. If you don't, then you can't do a perfect source separation. What will happen is that the strongest signals will go into individual outputs, but then it'll have mixtures of the weaker ones at the very end. Yeah?
What is the specific properties of the nonlinearity which makes it good
For super Gaussian, okay, it's the fact that the sigmoid, okay, has the same long tails that super Gaussians have. And that's one of the key things, is that you're using information in those tails. This was also generalized, by the way, to sub-Gaussian. So it turns out that if you have a signal, and there are like 60-cycle hum is a good example of that are sub-Gaussian, it will also be able to separate those. The only case it doesn't separate is when it's Gaussian, and there's a theorem that mixtures of Gaussians are Gaussian, and therefore there's no way you can separate it unless you have some other information, temporal correlation or something else. Yeah?
What happens if you apply ICA to clusters?
Ah, boy, this is a good question. Could you turn the lights off? We generalize it to clusters, and I'm going to come back to that. But first, I want to show you what Tony Bell did with this. Okay, he used this new algorithm to answer a long-standing problem. You can leave one of the lights on. I think if you just leave one of the lights on. Could you turn one light on? Just one out of the N? Okay, there you go. That sounds good. Is that okay? Okay. Okay. So now we're getting back to the brain. Okay. So the theme here is how can we go back and forth between developing new algorithms for analyzing complex data sets and using those algorithms now to go back to the brain and try to answer difficult problems in analyzing data from the brain. And then if we understand something about the brain, we might be able to go back and develop new algorithms. So let's go back to the brain. So this story began in the early 60s when Hubel and Wiesel described for the very first time the properties of neurons, single neurons, that were recorded at the tip of a microelectrode in the brain of a cat, visual cortex. And what they did was to record from a neuron and then show that by projecting patterns of light, that this neuron only responded to a very small part of the whole visual field called the receptive field, and that's this little square. And furthermore, it was selective. It didn't respond to any old spot of light. It would prefer oriented bars and lines in a particular orientation. And for this work, they received the Nobel Prize in Medicine or Physiology when I was at Harvard Medical School as a postdoc. So here's how they described the properties of a class of cells they called the simple cells. What they showed was that it was oriented, and I'm showing you here vertically, but all possible orientations are found. And they found that what they did was they said that if you had a line to the left here, you would excite, you would produce a burst of firing. But if it was to the left, you would inhibit the firing, right? And this was done all by hand. It was just more recently, within the last 1995, in fact, new techniques have been developed for automating that using reverse correlation and using flickering stimuli. But the answer that you get when you automate it is exactly the same that human weasel first got. So that they clearly were seeing something that's part of the architecture of the brain. Now, what Horace Barlow, a visual physiologist at the time, speculated was that these simple cells might be nature's way of reducing redundancy for natural images. That is to say, over the ensemble of natural images, if you looked at the correlations between these different simple cells, that they might be uncorrelated, independent of each other in terms of what they're representing about the visual world. At the time, there wasn't any way to test that because we didn't have any algorithm for analyzing natural images to find out what would be a good basis set, what would be a good set of filters that would do the automatic decorrelation and going further beyond that to make them as independent as possible. So here's an example of a natural scene, one of many images that we used. Here's another example of an image. You know, textures, rocks, all kinds of interesting structure there. So let's apply ICA to a 12 by 12 patch, and we're going to randomly take samples from these images and feed them to the ICA algorithm, which is going to now be 12 by 12 means 144 sources, right? So now what we're doing is we're trying to find spatial filters which are as independent as possible across this ensemble of inputs. And here they are. There are 144, each of these little boxes is one filter that turned out to be a subset, a set which are complete. That is to say, we can decompose any 12 by 12 square of any image into these 12 components, 144 components. And if you look at them, gray represents 0, black is negative, and white is positive. The majority of these have the form of simple cell, namely oriented excitatory and inhibitory regions at different locations throughout the visual field. There's one for DC, and there's some very low spatial frequencies, and there's some high spatial frequencies. These basically are, in fact, statistically identical to the properties of real simple cells. And so what this does is it confirms Barlow's conjecture. So we now think that at the very first stage of processing, natural images are converted into a format to make those signals as independent as possible for further processing stages. It simplifies the calculation by getting rid of all those correlations. Okay, many other applications. And now we're going to come back to the mixture model. You've already seen sound source separation, and I'm going to show you some examples from image processing. The same mechanisms, the same algorithm can be used for underwater communications. And the one that I'm going to focus on here to really try to drive home the point about trying to get to these brain signals is advances over the last few years of applying independent component analysis to analyzing EEG recordings. Those are the weak electrical signals on the scalp, and also 3D, or actually 4D data sets, 3 dimensions of space and 1 in time from brain imaging studies like functional magnetic resonance imaging. So, so here's a problem with where we can take the very same approach that we took with natural images now and apply it to text. And we're going to actually do this with a generalization of ICA, which is mixtures of ICA. You're all familiar with mixtures of Gaussians. So the idea is that you can either describe a little patch with one set of basis functions that were learned from this group or one from this group. In fact, the algorithm is smart enough so that you don't even have to tell it ahead of time whether you're taking it from here or from here. The algorithm automatically separates them out into two different basis sets and produces two different sets of filters that you're going to see. So here's I'm just taking small 12 by 12 chunks out. You can see this one obviously came from the right side, whereas these probably came from the left side because they have more texture, more linear features at angles, and so forth. And so we're just going to take randomly little 12 by 12 patches from the right and the left, feed them to ICA, and see what we get. So here are the two mixtures. One mixture looks a lot like the one we got from natural images, and indeed that was a natural image, so not too surprising. And here are the patches were represented by this set, but the patches that look like letters are represented by this set, and this set is adapted to and more efficiently encodes the features that are represented in letters. You can see much more vertical and horizontal end stops here that are very localized. So it's clear that these filters here are the ones that would be picked out to represent letters. So let's do that, and I'm going to show you that approach can be used to not to solve, but at least to help you get a leg up on a very difficult problem in image processing, which is scene segmentation. How do we take out, especially in a cluttered field, an object from its background? So here is a page from a Scientific American article. I can't read the actual March 1999, page 87, and this was a composite of a natural image and some text. And if you look carefully, you can see a little lizard here, which is camouflage, but it's actually sitting on top of the bark, right? So let's apply mixtures of ICA and see whether we can segment this into parts that have different significance. So one of the things you have to tell the algorithm ahead of time is how many mixtures there are going to be. Are there 2, are there 3, are there 4? If you tell it that there are 2 mixtures, then it separates the foreground from the background. In other words, the white stuff. Not too surprising. That makes a lot of sense. That's what I would expect to happen. But it treats the text the same way that it treats the bark. So let's ask it for 3 mixtures. So here's what it does with 3 mixtures. Red is mixture 1, green is mixture 2, and 3 is mixture, the blue is mixture 3. Whoops. I guess we have to start over again. Well, I'm sorry, I had to plug it in. Okay. So red is mixture 3. So the key thing here is that I didn't tell this algorithm anything about text or bark or backgrounds. It figured that out for itself just on the basis of image statistics from little patches, right?
And all you use is training was this one image.
It just used the one image, yeah. In fact, you don't have to use the whole image. You just use the top half and it will generalize to the bottom half. And you can't probably see it very well, but you see this little green patch here? That's the lizard. So somehow I felt that the lizard had a little bit more in common with the text than it did with the bark. That may just be anecdotal. I mean, I don't know whether to believe that or not. But in any case, if you what? Oh, yeah, it will separate out, but it'll probably do it on the basis of something that's not intuitive, like the barks of different textures or something. Yeah. Right. Right.
How did you do the segmentation? Or you don't?
Okay, how did it segment? Okay, so I skipped over that. So I told it there were 3 mixtures. What it did was it got a patch, and the first thing it does is try to decide which of the 3 mixtures is the one that comes closest to representing it. So some sort of error measure of how well it does. And then after you've trained it up, it will automatically assign each patch to one of the 3. So that's the segmentation.
Yes. But I thought I parallel process to some of these you have to hold them.
It's blinking. That means anything.
Yeah, it's blinking
But that's what I tried.
You did it at the other end.
Oh, I see, okay. Okay. Okay, so what else could we use this for? I mean, this is great. We have this terrific technique. There are a lot of practical problems in the world that seem trivial to you, but it turns out that are difficult to automate. One of them is barcodes, right? Barcodes come in many flavors. For example, the ones that you see in supermarkets, so this is the traditional linear barcode, but the post office uses the one that's a little bit different. And now there are a lot of matrix barcodes that you probably see on license, driver's licenses and so forth that carry a lot of information about your driving record, like whether you've been speeding recently. So one of the things that you might like to do is to have an automatic laser gun that doesn't have to be told which of the 3 barcodes it's looking at. Right? So let's see if we can segment them. We give the 3 barcodes. This, by the way, was a project that was done in collaboration with Symbol, which is the leading barcode gun manufacturer in the world. And it comes up with its classification into the 3 different categories and is able to accurately take each one of these barcodes and recognize, and then on the basis of the type, send it off, including junk. In other words, it's going to be looking at the address or something, and then send it off to the proper place to decode it. So, well, it's independent. No, it's a texture. You can actually feed it random orientations, and it will figure that out by itself. You can use it for denoising. So here's an image, the original image. We just simply added lots of noise to the pixels. Classical problem with Wiener filters. This is the optimal linear solution for denoising. You can do the same thing with ICA, and it preserves a lot of the fine edges here, getting rid of some of the noise in the background, smoothing, but not across contrast boundaries. Okay, now in the last part of the lecture, now I want to turn to the problem of EEG, and EEG has turned out to be one of the most difficult signals, although it was the first signal ever to be discovered about the brain, recorded at the scalp. They're weak microvolt signals. It's turned out to be incredibly difficult to understand what the meaning is of the EEG signals, and it's pretty clear why. If you're sitting here at the scalp, because the brain is a very good volume conductor of electricity, signals coming from all over the brain will contribute. And therefore, it's a massive mixture problem. But this is exactly what ICA is good at, right? So let's suppose that we're recording from 25 places on the scalp, each getting a mixture of 25 sources coming from different parts of the brain. Could we use ICA to separate them out and figure out what's going on in different parts of the brain? Now, to make the problem even more difficult, there are huge artifacts because the eye has a standing dipole, and every time you move your eye or blink, it causes a low-frequency, high-amplitude artifact as shown here, especially in the frontal leads, F7 and 8. Here's an EOG, which is over the eyes, so you can see when the eye is moving here. The way that neuroscientists get around this is by just simply cutting out this part of the record and only looking at the uncontaminated data. So so we applied ICA to that data set. And here is the components. Component number 1 turns out to be an eye movement signal. The eye is a perfectly good source, and it's a linear system. It figured out that the eyes are different from the rest of the brain. Okay, that's what one of the reviewers told us when we tried to publish this. And so we went and dug in and looked at some of the other components, and it turns out that some of the components that we were able to pull out actually came from the ocular motor system that drove the eyes. And we knew that because those signals occurred before the actual eye movement. So it's not totally independent. And here's another eye movement signal that is a vertical deflection of the eye. Here's a noisy lead that causes high-frequency noise. And muscle activity, it turns out that the jaw muscles produce a very, very nice high-frequency noise. So that by just looking at this and knowing something about the movements of muscles, we can actually identify what these are likely to be, and then simply, because it's a linear system, cut them out. So here's a cleaned up set of EEG. This is what went in. We cut out all of the artifacts. We simply subtracted them out, and here's the cleaned up data. And you can see, I think, in a way that would have been difficult otherwise, that, for example, here, on the frontal leads. You see, this stuff here would have been cut out, but look, there's actually some good signals there that would have been thrown out. And in fact, as a consequence of the fact that most of the artifacts are in the front of the brain, we know least about the EEG signals from the frontal cortex, which is one of the most important for planning and working memory.
Quick question.
Yeah.
You still name them the same way. So is this based on sort of the support of each component?
Yeah, so what is a component? Okay, a component, when you do the separation, you get two things out. You get out a scalp map, which is a fixed spatial filter. It's how you weight each of the electrodes. And then when you pass the original data through that electrode, you get this temporal waveform. So a component consists of a waveform and a scalp map. And you look at the scalp map, and that tells you something about what the possible sources are inside the brain. Here it's obvious it's the eyes, because it's in the front part of the brain, horizontally and vertically. It may be more difficult for some of these others because of the fact it may be buried deeply in the brain, and you really don't know where it's located, but you have a rough idea. Okay, I just want to show you this to give a sense for the richness of what you see there, and I'm going to give you a specific example. Here are 6 individual components, just to give you a sense for what the classifications are. This is an artifact which is clearly an eye movement artifact. Here is, and how do we actually display this? What I'm going to do, and this is really important, this is absolutely key. We're able to do the source separation on single trials. The traditional way of analyzing these weak, noisy signals is to do signal averaging. So typically what happens is you give a flash of light, a stimulus, and then you ask the person to press the button, for example, if it's a letter S or a letter H, right or left hand. And now what you do is you look at the sequences of deviations that occur after that, and you average over 100 trials. That's called the event-related potential. The event is the stimulus, and the potential are these wiggles that come afterwards. But by averaging it out, you're getting rid of all the phase information. So there's an enormous amount of information that you're throwing away. With ICA, we don't have to throw away that information. We can actually look at the actual waveforms of individual trials, and they carry a lot of information about ongoing oscillatory activity that's in the background. It's called the EEG. That's always going on, even in the absence of any stimulus. Some people call it thinking. So what we did was, first of all, this is a reaction time trial in which the person pressed a button when they recognized the stimulus. And the moment when they press the button is shown in black. And what we've done is ordered all of the trials from the bottom to the top, color-coding positive going signals in red and negative going signals in blue. So you can sort of get a visual picture of what this signal looks like for single trials, but averaging in this spatial sense. And we order them according to reaction time from shortest to longest. Now, if you just average all these signals, you get this average event-related potential, which has little wiggles in it. And you can see that this one is positive here, negative, positive, reflecting some activity going on at different time delays following the stimulus. But here's another component that is response locked. And you can see here that if you average over all the trials, you get some smeared out activity. But in fact, each one of those red areas here, which represents the positive going waveform, is locked to the response to the actual button press. And if you look a little bit before that, you can see there's a little negative going, which is also locked to the just before the button was pressed. But the majority, this was the minority of the signals. The majority of the signals actually were ongoing EEG signals, like what's called alpha waves, 10 hertz signals, that were going on, you can see, even before the trial began. And some of these were interesting because when the stimulus appeared, they didn't change the amplitude of the alpha wave of that component. Rather, it reset the phase, so that within 100 milliseconds, they were all, regardless of what the phase was at the button press, which is random, within 100 milliseconds, they were all now in sync. And that produces something that looks like oscillating potential here. There's another alpha source over motor cortex called a mu rhythm, which actually goes away when you press the button. It's called mu blocking. So there's a very vast dynamic richness here of signals. And I want to, and now we can actually use this almost as if it were a phased array system to actually go into the brain and pick out different things happening in different parts of the brain. And it's unbelievably interesting. Much more interesting than anybody could have imagined. And I'll tell you why people missed it in the next, after I show you this one example. So many years ago, people noticed that if you make a mistake in one of these tasks, especially if there's money on the line, there's a massive negativity in the frontal cortex. And some people call it the the error-related negativity. Other people call it the OSP, which it stands for the oh shit potential. And that's because the people who especially when there's money on the line, they're really trying hard, and often they're guessing, and they know before they've actually pressed the button that it's wrong, and they say, oh. And in fact, it reaches this negative point well before the actual motor response. So it's within the first 100 milliseconds that it reaches that strong negativity. These are the trials where you get it right, the averages, and this is the one trial where the averages of all the trials that you get it wrong. So you can see how different it is. Well, where's it coming from? What does it mean? Well, you know, this has been a big problem in the field. How do you go any further? It's known that in terms of the scalp map that it's focused here over the frontal cortex, but that's as far as you can go. You really can't localize things. So we collaborated with a group in Oregon, Don Tucker, and Scott McCaig in my group was able to analyze his data and find something in it that he suspected was there but had no other way to find it. So here's the task. It's called the Erickson line flanker task. It's a very simple one. The person is instructed to press a button with your right hand when you see an S in the middle, and press the button with your left hand if there's an H in the middle. Now, to make things a little more difficult, you put letters around it to confuse the person so that they're getting mixtures of S's and H's, and they're told to focus attention on the middle one. Now, to make it even more difficult, there's a deadline. They have to press the button before this deadline. They just can't wait and then think about it. And to really make it frustrating, you pick the time of the deadline to their average mean response time. That means they can't possibly beat themselves on average, right? But they're trying. It means they make a lot of errors. That's good, because we want to see what happens to the brain during the error. And then much later, they get the feedback, and they're given the money. So I want to show you some raw data, and this is something that you'll never see in any EE talk from an event-related potential person, because they don't show it because they think that it's noisy and it's not worth looking at, okay? So this is actually raw data that's been put through 2 scalp maps corresponding to 2 independent components, right? So all we're doing is some spatial filtering, and red is a spatial filter that has its, somehow the top, the peak is around the parietal cortex. You're looking down here on the person's scalp, that's the nose. And this one here has a peak, the blue one, somewhere over the prefrontal cortex in midline. Now, just look at a couple of these trials. Let's look at this one, for example. So you see that it's wiggling up and down. It's noisy. But occasionally there are these bursts where it looks like an oscillation for at least 2 or 3 cycles, and then it goes back into a lower frequency. That's called a theta burst. And that's because it is occurring at a frequency of about 5 or 6 hertz. It's the theta band in EEG recording parlance. See, here's another theta burst. Here's another theta burst, small. So what's going on is that these spatial filters, this ICA component, is picking out coherent activity amongst small pockets of neurons, one in the front of the brain, one in the back of the brain, that are very distantly separated. And they're both undergoing these theta bursts independently of each other. What we're going to be focusing on is the deadline here, which is the point at which the deadline occurs. The subject doesn't know when, the light doesn't come on or anything, but they figure it out after a lot of trials that it's going to be fast, right around here.
I don't understand. How do they get the signal? Because they're pushing on the...
Well, so the subject sits there, yeah. And then after a certain delay between trials, and one of these displays comes up, which has an S in the middle or an H in the middle. If the S is in the middle, they press the right button. If the H, they press the left button. But the signal, the visual signal, sets off a lot of activity in their brain, which then eventually leads to a motor activity in the motor cortex, which allows them to press the button. And we're examining that.
What's the deadline?
Oh, the deadline is chosen to be the mean reaction time.
But the deadline is because the image goes off?
No. No, in other words, well, first of all, you just have the person sitting there and you do 100 trials. And just figure out their average reaction time. Suppose that it's 350 milliseconds.
Okay.
That's how long it takes on average to press the button. And so now you tell them that there's going to be a deadline. I'm not going to tell you when it is, but if you press the button with the correct answer before the deadline, I'm going to give you $1. But if you press it after the deadline, regardless if you get it right or wrong, I'm going to take $1 away from you.
Okay. And then, do they
That's
get
the game.
Feedback?
And they get feedback later. They actually know.
Or they know that
Oh, after a second, a light comes on, bang, mistake. But they know when they make a mistake, yeah. But that comes long after the stimulus goes away and they've pressed the button, right? Okay, so the real question is what's going on here? Because this is when this error-related negativity occurs during the first few hundred milliseconds. So here is our best guess for where the frontal source is located. It's in a part of the brain called the anterior cingulate cortex. And it's known to be an area that gets a lot of information about how well the rest of the cortex is doing and whether the task is going smoothly or whether you're having problems. So it's a way of monitoring. It monitors how well the brain is doing. So it's kind of nice that the source was located in a part of the brain that neuroscientists thought there would be a signal, which was monitoring performance. The other signal turned out to be localized somewhere in the back of the brain, and here we're doing a dipole fit. We take the signal and we say, is there a single point in the brain where a single dipole will give us the best scalp distribution that matches the one that we see? And it's also in the midline, but it's in the back of the brain, distantly, very, very far away. And now we're going to ask the following question. We're going to say, okay, let's look at these two signals and ask when is it that the theta bursts in them become synchronized? When is it that become coherent? So what we do is we do, first of all, a map which is based on, this is time. Here's the stimulus comes on. This is frequency. So we do a map of coherence as a function of time and frequency. This is 10 hertz, 20 hertz, 30 hertz. And you can see that before the light comes on, there's a background EEG, which is called the alpha rhythm at 10 hertz. This is well known. It's very well established. There it is. So there's coherence, by way, between these two sources, which means that this is somehow, the information between these two different locations is somehow synchronized. Stimulus comes on, the alpha disappears, and then if the top one is when the subject gets it right. If the subject gets it right, then nothing happens until after the deadline, and then a higher frequency appears, which is in the 15 to 20 hertz range, and then after the reward returns to baseline, where you get alpha. Looks completely different now, or quite different. We look at the error trials. This is the case where the subject pressed the wrong button, and here's the case where the subject pressed the right button, but at the wrong time. And you'll see that what makes the difference between the top and these two is that there's a very large positive coherence between these two sources in the 4 to 8 hertz range, which is the theta range. So our picture now of what's really happening in the brain is that if everything goes smoothly, there's not a lot of communication between the front and the back. But if you get an error, suddenly these sources, there's a burst of activity in the 6 hertz range that's synchronized between the front of the brain and the back of the brain. It's like a communications channel has opened. And now there's information being exchanged. So the flavor of what we're able to see now using ICA and a lot of variety of other signal processing techniques is that there's a very rich communications channels that are going on at different frequencies, at different times in the task, under different conditions, under which those long-range white matter connections that connect the front and the back of the brain, which are very sparse, but those channels carry information that is synchronized. The information in them, or you shouldn't say information, but the signals in them become synchronized so that the two parts of the cortex may be able to communicate more efficiently. Now, we don't, here's the bottom line. Over the last 50 years or so, neuroscientists, primarily by recording from one neuron at a time, have been able to plot out how neurons represent the world. How, in Hubel and Wiesel's case, how a single neuron in the visual cortex responds to a particular pattern of light, an oriented bar. That's the feature it likes. People have repeated that experiment over and over again. They've looked in the front of the brain. They found parts of the front of the brain whose activity is elevated during what's called the delay period, when the animal is trying to remember where to move its eye. That delay period is a representation of where the animal is planning to move its eye. But again, it's the same, asking the same question. What does the neuron represent about the world, about my plan, about an action in the motor cortex? Representation, 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 right? Even though they've been putting the electrodes in different parts of the brain, they've been asking the same question over and over again. Well, here's a completely new type of question that we could begin to ask about the brain. Let's not ask what that area or neuron represents, but let's ask how that information is communicated to other parts of the brain, and how is that regulated? What determines when two areas become synchronized? What determines when that information flows? I think that if we really think about this from the perspective of a complex system that has a huge memory and is organized in a very non-intuitive way, we're faced with a really difficult problem. So I can tell you, for example, I can sit you down and say, I want you to press the button with your right hand when the light comes on in the upper left-hand corner that is a red color. Okay? So fine, I sit you down. Every time the light, I'm going to have distractors. There's going to be some green lights going on and some red lights in other parts of the visual field. But after a while, you're 100%. And every time the red light goes on in the upper left-hand corner, you pressed your right hand. Okay? Now, if you think about it, where's the information about the red light? It's located somewhere back here in the back of the brain representing the visual world, but only a small number of neurons, because there's a lot of other neurons representing different things in different parts of the visual field. Somehow, those neurons have taken command of my right hand, represented by a bunch of neurons, a small number of neurons in the motor cortex that press your right hand. There's no direct connection between them. How is it that you got that information there, right? It's not a hardwired network. I could have told you to press the button with your left hand, or I could have said press the button if it's a green light. It could be any arbitrary sensory input mapped into any arbitrary motor output. Not quite arbitrary. Some are easier to do than others. But the key thing is we have to have a flexible communication system to be able to reconfigure it so that you can do that on the fly and in a way that allows us to be much more adaptive and flexible in our behavior. And that's something that humans in particular seem to have evolved to be able to do extremely well. I think I'm going to stop the talk here because I've already gone on longer than I expected. If you're interested in brain imaging, it turns out the very same techniques that we've used for analyzing EEG can also be used for analyzing the brain, the blood flow signals that are found in the brain. Let me just quickly jump to the end. Just to say, first of all, that there are a lot of people, there are over 300 people in the world now working on independent component analysis algorithms. There are about 12 that have various strengths and weaknesses. Some of them based on cumulants, some of them based on other higher order measures. But the key thing, though, is that we've solved this classical problem of blind source separation. It can be applied to many, many different practical problems. We had an international meeting in San Diego in 2001 where 350 people showed up, and now there have been 4 of them, the last one being in Japan, the next one coming up in Spain. There are 3 heroes that helped develop this algorithm. First, all from computer science, Nikki Shroudoff, who joined the lab from UCSD and actually pioneered the application of entropy to this class of signal processing problems. Paul Viola, who was a graduate student at MIT at the time, but decided to do his thesis with me. So he came and spent a couple of years. He applied these entropy methods to doing image registration, for example, between 2 different modalities like CT and MR, which is now the standard algorithm used in medicine. And then finally, Tony Bell, who is the person responsible for the Infomax algorithm. So thank you very much. Could you flip the lights? I think people have already asked a lot of questions, but maybe if you want to stay around, I'll answer a few more. Yeah.
I would like to come back for a second to the natural image, which is used by taking randomly some patches well by well. Assume that you will have a bunch of images trying to cover statistically all the natural images, and you will have thousands of patches, and you will apply ICA and retain, say, for the sake of let's say 256 independent components out of these patches. Do you imagine that we could eventually design a new image compression system using this?
Formally, what's going on is that the number of bits of information. Okay, let me just back up a little bit and explain a key concept here. Okay. So in a communication system, what you're trying to do is to transmit a fixed amount of information through a channel using some words. The question is, what words do you want to pick? Well, it turns out that the words you should pick are the ones that are adapted to the particular class of images or speech waveforms that you're trying to send across, right? So it turns out that for a lot of, what would be the ideal thing? The ideal thing would be if you had the words that you picked were organized in such a way that you only had to send across a few bits to tell which word was the key word for that object that you had. That's called a sparse representation. The worst would be if you had to send a number for each of the words, like it's a half of this, a third of that, and so forth. In the case of natural images, if you use sines and cosines, then you're in a distributed representation where it's not very efficient. But if you use ICA, then it becomes very sparse. That is to say, you only have to send over information about a few of those filters. So the sparsity turns out to be a very strong result of independence. In other words, independent channels means that if one guy is on, he's saying, this is my stimulus, I can really represent this really, really well. There aren't a lot of others that are going to be activated. And that means you only have to send information primarily, if you threshold it, about one or a few stimuli. And so this turns out to be a very natural way of finding getting a set of filters which matches the statistics of whatever it is that you're trying to use as your inputs. We've used it, for example, on faces, which is kind of cute because you can very efficiently represent faces by little independent filters that represent eyebrows and eyes and mouths and so forth. So it's like Mr. Potato Head, which is a very good way, if you think about it, that if you're a cartoonist, that's exactly what you do, is you only have limited amount of information that you're going to pencil in, so you put in the parts that are carry the most information, and that's what's called a sparse or independent representation. Yes?
So I have a question about sparsity, actually. When you, let's go back, actually, this is the easiest example, I guess, to the X that you showed.
Right.
Where PCA does the wrong thing and ICA does the right thing. So there's more that you can say. I mean, for one thing, just to check my understanding before I go on to the question, so stop me if this is wrong, is you have these two mixture components. Now, you didn't show the coefficients of the mixtures, but if you had shown the coefficients of the mixtures for each point, the first point is knowing the coefficient, knowing one coefficient doesn't give you any information about the other coefficient. That's what ICA does.
Right.
But the other interesting thing is that when the coefficient of one is large, then the coefficients of the other tend to be small. Right? So is that right?
Right.
So there's a way in which what you're looking at is something that's a little more like a mixture there. And that's also the case for some of the other examples that you showed, I think, so you were just talking about eyes and mouths and noses. Well, when you've got a mouth, probably that patch is not also showing a nose. So you would expect that the representations in the transformed space would be sparse vectors or close to sparse vectors.
Exactly.
Yeah. So are there versions of ICA which take that constraint into account?
Okay, so the result is that we have shown that under the condition of the assumption of independence, that you automatically get a sparse representation, right? And a sparse representation minimizes the coding, it maximizes the coding efficiency, right?
I see. Okay. So you get this for free.
Right, exactly.
Oh, right.
There are other people, in particular Bruno Olshausen and David Field, who went about it the other way. They actually started out with the assumption that let's look for a sparse representation. And interestingly, for visual natural images, and they converge on exactly the same set of filters that we found. So it's a wonderful convergence that these two principles seem to be consonant.
Great. Thank you very much.
Richard.
Back to the number of microphones and the number of speakers. You have so many electrodes on the scalp,
Right.
but how many different places in the brain are sending out signals independently?
Right, very good question. So how do we know how many sources there are? I mean, this is a really fundamental limitation that we have. And the number you need is really determined by the signal-to-noise ratio of the signals that are being picked up by the electrodes. We're now up to about 120 electrodes. And so one way of solving the problem is just keep adding electrodes, right? And find out when you stop picking up signals that look like they carry information. And that's very obvious because it turns out the scalp maps, which for the big signals, they look dipolar. When you get up to the 30th or the 40th, they start looking like random noise, like snow, suggesting that there's no coherent pattern, spatial pattern, and therefore you've probably lost all the signal. So from our best estimates, they're probably, the ones we can pick up with our current system, is probably about 40. And they're, it's not the same for every task. So if I put you in task A and compared the components I get from task B, they maybe have 20 that are in common, and there may be 20 that are specialized for the task, suggesting different parts of the cortex are in fact taking part in solving different tasks, which is good, because that's really what neuroscientists have discovered over the last 100 years, is that there's a principle of localization in the brain. Different parts of the brain have different tasks that they take care of: auditory, visual, motor, and so forth. So, but we're still at a very early stage. In fact, in order to be able to prove this to you, what we really need to do is to simultaneously do ICA on the EEG and have the person in an fMRI system and find out where the activity is located using this 3D method, functional magnetic resonance imaging, and then somehow coordinate that with the sources for the ICA. And so we're right in the middle now of doing that. We've solved a lot of the technical problems, and it looks like we're getting some good results. Yes?
The actual way in which humans solve some of these separation problems, however, must be a little bit different. So, for example, in the case of the tree trunk versus the text, I mean, it's also just the fact that people have prior knowledge of these objects, so they're just matching the input with prior knowledge.
Well, what does prior knowledge mean?
Right.
What does that mean?
That was going to be my next question.
Okay. So here's my guess at what prior knowledge means. It means that your brain has created somewhere neurons that respond to text and not to tree trunks or bark, okay? But there may be another part of the visual system, higher order in the visual system, that recognizes trees and faces, right? And so that's exactly what we think is happening, is that over time, your experience with the world may allow different neurons in different parts of the visual system to become experts for that class of stimuli, especially something like reading. It's very overlearned where you're doing it all the time, right? They're arbitrary symbols, but once you've committed to them, you really have to sort of, it becomes automatic. Right. Right. Yes?
So in terms of the question about what independent components mean in the EEG, one question that occurs is whether those things are localizable. I presume you do dipole localization.
Right.
So do these ICA independent components localize reasonably?
Oh, yes, yeah. So part of the reason why I have some confidence that ICA is just not giving an arbitrary decomposition. For example, when we use PCA on the same data set, we get basis functions that look like sine waves, right? And I don't think for a minute that those have anything to do with what's happening in the brain. It's just a compact way of representing the variance in a few components. But the reason why I think ICA is giving us something more than that, something that is more than just an arbitrary decomposition, is that we never told the algorithm where on the scalp the electrode was, right? In other words, we could have numbered them completely differently. The algorithm doesn't know. It's given one vector at a time, 120 electrodes, 120 numbers. It doesn't know where they're located. The fact that at the end it finds a component that has a smooth scalp distribution has to reflect something in the brain itself. I don't see why else it would have been smooth. And not just smooth, but can be fit with 95+% variance to a single dipole, despite the fact that we know that the scalp introduces some nonlinearities and spread of the current. Even just with a single dipole, assuming a uniform medium, we can fit 95% of the variance to most of the dipoles. Sometimes you have to have bilateral dipoles, say in the back of the brain for the visual system. But that really does, I think, that's a good sign that this is a biologically relevant decomposition. But again, until we actually have ground truth, some way of comparing the ICA with some other measurement. Another way to do it would be to put in, and this is something that we're planning to do in collaboration with some doctors at the Mayo Clinic, is during operations in which they're trying to localize epileptic seizures, they put a grid of electrodes onto the scalp. And there you can pick up local field potentials. And so we could do an experiment in which we record these local field potentials from the brain itself at the same time that we're doing the ICA on the scalp. And the question there is, because you can get much more localized activity from the local field potentials. It'll tell you within a millimeter of where the signal's coming from. And there we'll be able to compare the inside of the brain and the outside of the brain. Amazingly, nobody's ever done this before. People have been putting these grids in here at Hopkins for decades. I mean, they're doing it 15, 20 years ago. But no one has done this experiment. And part of the reason why no one has done it is that EEG fell out of favor. No one really cared about it because it wasn't telling us anything about representation. And now I think we can go back and look at the signals, use modern signal processing techniques, and really begin to mine it for what I think is really represented there is not the information itself, but rather the way that the brain encapsulates information and reorganizes itself in an online, on-time fashion to be able to get the right piece of information from one part of the brain to another part of the brain so that you can make the right action. Yes, Paul?
Yeah, this is probably just a simple conclusion, but if the ultimate conclusion of the kind of study that they're looking at for the OSPs is that you're detecting a kind of synchronization among distant sources, if you heard that at the outset, then you might think that what you want to do to the signal was to look for components that were maximally non-independent. And so why is it that by looking for components that are maximally independent, you end up finding out that these two sources are tightly coupled?
Very good question. Okay. So, so if you look for things that are correlated, right, you'll get what you get is what people already know about, namely this part of the brain is connected to that part of the brain, and there's a strong coupling between them. By asking the other question, looking for maximal independence, we've shoved them apart as far as we can in signal space, so that they're doing things at different times in the task or for different types of tasks that are different, as if there were two people talking in a room about two different things, or two instruments in an orchestra that may occasionally be following the same beat, but actually have very different scores, right? So the following the same beat is the correlation. So if you went for the correlation, you'd find out, well, there are a lot of different instruments playing the same beat. But what you really want to do is find out, okay, well, what's different about them? What is it that they're telling us that's different? Now, having the algorithm actually, if the sources are not independent, the algorithm obviously is not going to separate out independent sources, but it will try to get them as maximally independent as possible. And what you can examine then is the residual. And the residual are these weak, transient, fluctuating coherences that are on top of those signals, right? So in a sense, what we're doing is we're looking for higher order statistics that are not independent, but nonetheless can be picked out different again, the fact that it didn't have to be in frequency bands that have been identified by brain scientists. The theta band is something that's very well established for memory problems, for example. There's another band that I didn't talk about called the gamma band, which is thought to be important for things like attention. And we can see all those signals. We can see them. We know they're there because people have measured them, but nobody knows what they're there for. Like the gamma band. There's a big story. I can give another lecture about this. A big story about recordings from neurons and showing that there's occasions when neurons become synchronous when they fire at the same frequency at 40 hertz. And for a long time, people thought this may have something to do with perception, that when you become conscious of something, the neurons begin to oscillate together, right? That was the first hypothesis. That's wrong. All the evidence is against that. First of all, you get these synchronous signals the strongest when you're unconscious and you're asleep. Second of all, they come and go, which means that when I look at you, Paul doesn't come and go. But maybe there's something about the signal that's important, this coherence idea. And so the hypothesis that we're pursuing now is that this coherence is an internal communications signal that the brain uses for attention, for taking a piece of information and enhancing its impact downstream. Because if you synchronize spikes, then they have more impact on another neuron by virtue of the convergence of spikes that are occurring at the same time. So that boosts the power in the signal that you're trying to attend to. But that doesn't change the meaning of the signal. The rate at which the neurons are firing is the same. You're only changing the spike time. So I guess what I'm saying is that we're now in a position to look grossly at the whole brain, to look at global communication signals. It's as if you were discovering what the brain's internet protocol is, but it's still very crude, it's very global. And then now we can go in and record from neurons, we can record from local field potentials, and work out the microstructure of what the packets of information actually look like. In the theta range, for example, the packets are spaced pretty far apart at the peaks of the theta. But in the case of gamma, where you're paying attention, they're spaced more closely together. They're separated with, for a 40 hertz signal by 25 milliseconds. So there are going to be many different types of coherent signals that mean different things in different parts of the brain. And we're just beginning to explore that. There's a whole new realm of neuroscience, which is a more global kind of organization than the tiny single neuron level that we have really been focusing on over the last 50 years.
So let's thank our speakers.